Hello, my name is Prateek. I am a research assistant professor at the University of Minnesota, part of the Galaxy P team, wherein we develop proteomics tools to be deployed within Galaxy. The team at University of Minnesota, led by Professor Tim Griffin, basically works on adding tools and workflows for proteogenomics research as well as metaproteomics research. And we have been doing this for last 6 years. We conduct tutorials as well as we have started using these workflows for multiple research projects. And I'm very excited to be part of this cancer proteogenomics workshop here in Mumbai, basically because of the fact that that's the research that we work on and gives us an ability to reach out to the audience here in India as well as the rest of the world. Right, so I'll try to answer this question in two parts. The first part that you mentioned was about a common platform, and that's exactly what we do with Galaxy platform, and I'll be giving a talk very soon about that. But Galaxy platform basically helps both genomics, transcriptomics as well as proteomics researchers to use a common tool or platform to use it. But in general, I mean, one of the main barriers that I see is most of the times researchers or developers are kind of specialized in one field and not the rest of them. And hopefully a platform like Galaxy or anything else helps to bring that common playground or a common place wherein all these researchers or developers can develop tools and help integrate the data. I think there is also a need for researchers to understand the fact that developers and users need to work together, because things that are developed by a developer might or might not be useful to a user. So it could be a great algorithm, does fantastic things, but if it is not something that aligns with the question that is asked by the researcher, then it just becomes an academic tool, right? Vice versa, the user also has to understand the possibilities and challenges that a developer faces and try to achieve tools that work and give you a multi-omic or a systems biology perspective to the data. One of the observations that researchers have started making now with multi-omics or transomics research, as it's called, when we're comparing, let's say, transcriptomic data to proteomic data, at least in early days, one found that the correspondence to each other in terms of quantitative expression was not exactly 100%. In fact, the concurrence was much, much lower. And that was a little bit of a concern earlier, but now it is understood that the way RNA expression works or protein expression works is not exactly instantaneous, in the sense you could have RNA expression and the protein expression could lag behind, right? Or you could have the stability of your RNA molecule determining how much of protein is going to be expressed. So I think it is really important that researchers start undertaking temporal or time dependent expression studies for both transcriptomics and proteomics to kind of make a much more studied conclusion on the expression of both protein as well as RNA. Because if you find that the RNA is low and protein is more, it doesn't mean that it's just giving you a particular snapshot and not the cycle of that particular expression. So I guess the answer to that is time dependent studies, and the technology is getting there. I mean, RNA-seq is already there, and I think with newer developments in mass spectrometry, the scan speeds are getting really fast. We hope that we can get deeper as well as a lot more data from mass spectrometry data, so that one can match that with transcriptomic data. I have a little bit of concern with that, given that and again, personally, I haven't really studied that as much. I've worked on domains and I've looked at using domains, how one can predict the function of a protein. But in terms of interaction, either one do not have enough information, or secondly, if you have predictive models, and if these predictive models are backed up by experimental data, then yes, one can say that. But until we actually have a good correlation between those two, I think experimental data is going to be a lot more dependent or a lot more determinant of what actually interacts with each other rather than computational modeling. But that's my opinion. I think it's important to start with RNA-seq data, and I'll be covering a little bit of that in our in the talk that I'm giving today. So if you start with the RNA-seq data, obviously you have your genomic coordinates, or you can go back to your genomic coordinates. And if you use that as a template and transfer that information to your proteomics data, or at least have database schema to kind of go back and find out your genomic coordinates or gene-centric approach to that, I think that's possible. We have shown that, and obviously there are tools and workflows that need to be first developed and then optimized and made robust enough so that one can do this on a more consistent basis, not only for known organisms, but also for organisms that are getting sequenced. But it definitely is possible going through the RNA-seq data. I think for the proteogenomics field to develop, one would actually have to make this almost a requirement, because if you do not correlate your protein to your DNA or to your RNA, you're almost losing that information, and you want to maintain that because you kind of know it's coming from from DNA to RNA to protein. Just the fact that if you don't have the tools available or the coordinates available is not a good enough excuse to lose that information. So I think it's going to be necessary as the field of proteogenomics becomes a more established field as it's emerging. So I have basically worked on two workflows or two areas of research. One is proteogenomics. So when we started working on Galaxy, or using Galaxy for proteomics, what we did not want to do was just develop another platform for proteomics research. Take in your mass spec data, your protein FASTA file, you get peptides and proteins. There are many tools which do that. What we wanted to do was take some challenging areas, and this was 6 years ago. So proteogenomics was very new, metaproteomics was very new, or I wouldn't say they were very new, but they were emerging, and we saw a promise as well as a challenge there. So what we did there was we worked with the post-processing of the peptides identified and trying to make it easier for a user to use it. And that's where, the challenge there is working with a user or a project and a developer. And as I was mentioning during the break, a developer is extremely enthusiastic about his work. The user is very focused on his questions asked, and sometimes these do not meet, and that leads to a program or a workflow which is great in its own field, but it's not usable, right? So I think the developers and users, if they work together on a project with the specific questions in mind, and then creativity starts coming in once you have the basic blocks in place, I think that's how tools are going to develop. In terms of its current status, I think it's in pretty good shape. I would say MZML format, MZ-identiml format are kind of getting accepted. The only part I think I see a need for development is the MZ-quant, or the quantitation portion of the MZML format. And I know there are developments taking place there, but I think making this more robust and optimized is going to be the need, because there I think are going to be many quantitative studies, and especially quantitative studies that correlate to RNA-seq data or any other quantitative analysis data. So getting the quantitative portion of the proteomics or mass spectrometry data is going to be important. I'll again answer this in two parts. One is, you want to develop something to show it works, right? And we have been doing that. We take small data sets, generate workflows, develop it on a cloud, build a Docker instance, share it with the world. We give presentations saying this works. But that's almost like playing a sport in a small little, backyard or something. The real data that comes out is not going to be just few raw files or a few FASTQ files, but it's going to be many and maybe multiple replicates and hopefully many time points as well. So you need to have something at the back end. The infrastructure needs to be such that it can support that. But it's also important that the tools and the workflows can run on that. They use the ability to use the vast resources that are available either in the cloud like Amazon and Google, or any supercomputing infrastructure that you might have. So I think it would need to go through steps. You need to make it work first, and then just like a child develops, you want to see that it graduates from that it's school to college and maybe into the real world. So there is definitely need for that. In academic settings, sometimes it's not possible because teams get funded for 5 years and then focus changes. But I hope the field in general kind of understands that and makes it possible, because it's really not much fun to go back, develop a new workflow and start again and again. So hopefully there is, this momentum keeps going on. At multiple levels, definitely the ability to ask good questions, and that doesn't come easily, right? I mean, some people are naturally talented; they'll ask good questions after really early in their education. But one thing that you developed as you do your advanced studies is you start asking really good, sharp questions. I mean, as a young student, you always have 10 questions, but you're not able to decide which of these 10 are good. You think all 10 of these are good, right? So asking good questions, and then secondly, designing experiments. It's always good to have great ideas, but to put it into practical, stepwise manner is important. The third part is sample preparation. I work in the bioinformatics area, and I know that many researchers kind of take it as sample preparation is going to be good, or you either blame the sample preparation or you kind of taking it for granted. But I think there is a lot of quality control that needs to be done, a lot of things that need to be considered. And that's true for data acquisition as well. You need to have QC parameters to ensure that if you have replicate generated on day 1 and generated on day 10, you can compare them. Or if you cannot compare them, what is the reason you cannot? So the measures to have that, and there are tools in place to do that as well. But I think most importantly, it's important that the researcher gets to interpret the data, right? And interpretation could be on various levels. It could be by using programming, or it could be just a biological interpretation wherein saying, good, I've seen this data, I know what it means, but I need tools to do that. And that's where the person can work with the developer, which is what I do. I work with developers because I kind of get a sense of where the data is going or what could be important. But you can start in one area and then develop into any other area, or you could just become an expert in one area. And then, once you start having that ability to look at overview of the project, asking good questions and publishing good science, then I think you've achieved the ability to do things. While I was answering, one of the things I kind of learned during my career was also ability to communicate. And this is not just through manuscripts and through through anything that's public, or I think you need to clearly mention to the person you're collaborating with, because team science is going to be very important now. You cannot be an expert in mass spec and an expert in, and you might be, but you might not have time to do that. So you need to communicate your expectations to your collaborator and get the best out of them, and also offer the best that they want from you. So I think communication is very important. I think obviously the younger generation already has kind of a strength in that because of the amount of social media that's available. But I think it's important to have effective communication while avoiding the noise. How do I get this across to somebody, which is a signal that could be useful, rather than giving you 10 pages of data and say, go find your answer. So I think these are a few things that develop, and I'm sure there will be new skill sets that will come up as the field develops.
Hi, my name is Ratnarajesh Tangadu. I am from a company called ESAC. We work closely with the National Institutes of Health, USA. So we are a bioinformatics and health IT company. We do provide a lot of services to both the National Institutes of Health and also the Office of National Coordinator in the USA. I think we can start with the sheer volume of the data. And then there is lack of resources and also the infrastructure to manage that volume. So everyone has all their kind of tools and the processes and pipelines, but not to handle the big volume of data. And the other thing I would stress is the lack of data standards or the lack of adherence to it. So what happens is you have a large amount of data, but there is no data standard, so you can actually you miss the ability to actually combine that analysis with other data sets that are pre-existing or that are coming from the other programs. So I think these are the main three things that strikes my mind to begin with. Data standards are basically a set of rules or agreed upon rules so that you represent your data in certain way. So annotate that and also represent it. So that actually helps the data harmonization part. So what happens when you start generating data which is specific to a particular program or a particular country or a particular disease type or particular population, and when you want to actually integrate the data into a bigger platform and bring the data from other platforms. For example, you call the same disease or the same gene by different names, and you know that it is the same, but the computer doesn't understand until you tell that. So data standards actually help you get to that point. And the other point is in the data harmonization, so you analyze all of the data through a particular pipeline, so that allows you to see all the data through the same eyes. So even though you can analyze each of those data sets independently with a different pipeline, harmonization actually brings them together and helps understanding on a larger scale. Earlier I mentioned about the lack of infrastructure or the lack of resources. So cloud computing actually removes that barrier. So it's on demand, pretty elastic. So there are a lot of companies out there which are pretty established, something like Amazon, Google Cloud, and now Microsoft Azure is there. So for that you don't need to have a data center on your premises. So you don't need to have a set of IT crew helping you out, adding more disk space and networking and all these things. So what you need is basically a good internet connection. Based on the volume of the data, it will scale up pretty quickly. So you can increase the size of the disk space that you are using and also kind of resources, so the compute power that you need. So it's pretty helpful in that sense. So in the precision medicine, so the question is specific to that. So what happens is, like, it's not the data about a particular program, but it's data about the individual patient or individual candidate or a subject. It depends on how you call that. So within the personalized genomic space, so every day the data is growing exponentially. So the Moore's law, it doesn't apply anymore. So cloud computing comes into picture to handle that kind of data. I don't see it as such a big problem, because the connections between the genes and the proteins at the level of accession numbers between The most common databases out there are, for example, RefSeq and UniPro. They are very well annotated and they are very well maintained. And most of the proteomic pipelines are actually rolling up the final protein parsimony results to the gene level. So it's pretty easy to actually map back to the individual isoforms, and also if you start from the isoform, you can easily come back to the genes. So now the big buzzword everywhere, at least at the level of the governments that are involved, the US and I would say the European Union, is the big data and the data commons. So data commons actually brings together everything at one place. So the resources in terms of the storage, the tools, the compute power, everything at one place. That's called data commons. So what the user needs is simple login. So like the way you log into your email account, so the user can actually just log in and he doesn't have to bring anything to the table. And the only thing that's there is he can take back some results from the analysis directly from the cloud computing platform. Like I said earlier, data standards. That's very important in achieving the goal that we have in front of us. There's the precision medicine, the large volumes of data, and so the siloed nature of the data actually never helps. So you have this genomics, proteomics, imaging, the immunology, all these data are sitting there side by side, but they can't talk to each other because there are no standards there. Let me take that back. There are standards there, but they're not adhered to. So what happens is you're calling the same thing with different names, like I said earlier, so you cannot actually do the integrated analysis. Keep your eyes wide open because there is a lot of open source data already available. So you do not have to actually generate the data. You can start looking into the existing resources, bring the data onto your laptop and start analyzing that. So to analyze, the simplest tools I would recommend currently are R and Python. So they're pretty easy to learn, and all the public data that's available there, you could process that data through those kind of parsing tools and the statistical packages, and we are ready to go. There are some portals that I know of which collate all the data from the literature, and also the Nucleic Acid Research is a journal which publishes the available databases and the tools on a yearly basis. But at the same time, now it's a requirement with most journals that you make your data available somewhere on a public repository, and also the tools that you have used, and also the versions of the tools. So it actually a lot of things that are already there that provide the metadata of the analysis that's performed on the particular data set. So the data is available, the tools that are used are available, and the versions are available. So it actually helps to reanalyze the data on a different settings or on a different data set, or just simply reanalyze and to see, cross-check the validity of the reports from the publication. That's critical to all of the data commons efforts that we talked about, right? So if you do not share the data, so there is no data commons. So if you want to share the data, that's always welcome. And now the governments actually require you to, as long as the research funding is coming from the public money, they are required to submit their data to one of these resources. For example, in US, there is something called Genomic Data Commons, where they make available all of the genomic data, and now they're building a proteomic data commons that we are building in our group. And then the other point is, so it actually helps innovation. So sometimes you start with a small data set. For example, the TCGA pan-cancer analysis is one such an example. So there are about 30 plus different cancer types that they have been analyzed. And now after so many years, the program closed I think at least 3 or 4 years ago. But now there is research going into that. And just because the data is shared, everything is available in the public domain, and people are using the data. So a lot of proteomic data is it's not protected. It's in other words, it's open access. So that's a good thing. But if you are looking at specifically at the proteogenomic kind of integration, so some of the genomic data is actually controlled access. So there are data access committees. So you need to submit an application. They will review your application, and there is no cost factor involved here other than your research interest. So so once you submit that, so they will review your application and see the research statement, and they come back to you with their decision. And there is the expectation that you adhere to the guidelines proposed by those kind of repositories. So what happens? This is all patient related data. So patient privacy is primal to all of the data sharing.